Okay, welcome to lecture 5, I believe this is. Today we're going to be talking about cellular anatomy and physiology. So, we're going to be kind of zooming in upon the tissues and looking at some of the fundamental organelles associated with the cells and also their functions within the cells themselves. So, a cell is a unit of life. We have about 75 trillion cells that interact to form dynamic tissues, organs, and organ systems. Cells vary greatly depending on, their function, their size, shape, and content. So today we'll spend some time talking about, not only some of the common organelles associated with our cells, but also the way in which substances can cross through cell membranes and allow us to have movement and communication. Most cells have 3 common components. Generally speaking, they're going to have a cell membrane, a nucleus, and a cytoplasm. And cells can also have specialized organelles, and an organelle is a specialized structure that can move and interact within the cell. Usually, again, we can have items that flow across the cell membrane, and this is going to not only allow us to have communication amongst the cells, but also allow us to complete metabolism and metabolic processes. So let's start our conversation by talking about the cell membrane itself. And the cell membrane is made of a phospholipid bilayer. Okay, so what that means when we say phospholipid is we mean that phos means it's got a phosphate, and lipid means that it's got a lipid or a fat component associated with it. Okay, so when we say a bilayer, you can see here that we've got one layer here and one layer here. So together, these phospholipids kind of line up next to each other and form a bilayer, or bi means 2, so 2 layers. Again, if we look at each of these individual structures by themselves, you can see that little yellow head is the phosphate portion, and it's got 2 lipid tails that are going to poke in towards the center. Okay. Our cell membrane, some of the specific characteristics about it, it's considered selectively permeable. And when we say selectively permeable, we mean that it's selective about what it can allow itself to permeate through. So, certain things can permeate across this membrane easily. Other things are not allowed to pass through. So oxygen and carbon dioxide generally can move through very easily, whereas other items need a specialized ion channel in order to move through. So we've got various types of receptors. So this is one example of a receptor that's located on the cell membrane. This would allow us to have some level of communication amongst the cell and with other cells. And then, like we said, we also have some various ion channels that are generally made of proteins, and that will allow certain substances to flow in or flow out very easily. Okay, like we said, the cell membrane is made of this phospholipid bilayer that is both hydrophilic and hydrophobic in nature. If something is hydrophilic, it means that it is water-loving. And if something is hydrophobic, if you have a phobia, you're scared of it. Hydro means water, phobia means scared, so it's water-fearing. So if you can kind of take a look at the previous slide, we have that phosphate head and lipid tails. This lipid tail portion is the water-fearing portion, that's hydrophobic in nature, and this head portion, this phosphate, is hydrophilic or water-loving, which is why the phosphate heads face outward, because they will be submerged in some type of fluid, and these tails are going to be hidden together, and again, that will hopefully prevent them from being exposed to water because they are water-fearing. Some of the functions associated with a cell membrane, let's see, we have 4 of them. One would be to allow movement of molecules in and out of the cell. Number 2 would be signal transduction. And signal transduction basically allows us to have communication between cells or between various parts of the body. Number 3 would be adherence to other cells, and we've got some kind of specialized structures that would allow that to happen. And lastly, the last function of the cell membrane is to form a boundary for us. Okay. So we've talked in some detail about kind of how this phospholipid bilayer has been formed. So again, you know that these phosphate heads are hydrophilic in nature, so they're going to face outward. We've got these these fatty acid tails, which are going to face inward, and those are the hydrophobic components. And because this membrane is selectively permeable in nature, we have various gradients. Okay, so we have a concentration gradient, and we might also have an electrical gradient. Together, these can sometimes be called the electrochemical gradient. Okay, so the combination of these two together forms an electrochemical gradient. We can have a concentration gradient separate from that of an electrical gradient, but a lot of times they are combined. So we're talking about a concentration gradient, kind of in this first picture up top. We're looking at a difference of the number of molecules on one side of the membrane in comparison to the other side. So for example, if we look here at these little yellow molecules, which are sodium in this picture, you can see that the sodium ions are more plentiful on the outside as compared to the inside. So we have a difference in the number of sodium molecules outside of the cell in comparison to inside the cell. And that would create for us a concentration gradient. We also have an electrical gradient in this image, and you can see that the cell is more positive on the outside in comparison to the inside. The inside is more negative in charge compared to the outside. Again, in this particular situation, that could be directly related to sodium. So you can see that there's a lot of sodium ions on the outside of the cell. Sodium ions are positive in nature, so maybe that would give us more of a positive charge on the outside of the cell in comparison to the inside where it's more negative. Again, the combination of these two factors together would create an electrochemical gradient. Okay, so we've spent some time talking about the cell membrane, how the cell membrane can be selectively permeable, the phospholipid bilayer, and how it contains both hydrophilic and hydrophobic components. And now we're going to move forward and talk about the cytoplasm. The cytoplasm is a gel-like material in which organelles are suspended. You could think of this as kind of like the jelly or the background within any given cell. So most of our cell activities are going to occur within the cytoplasm. So again, the cytoplasm is all this light teal type of stuff. It just basically forms the background or the backbone for the cell itself. Cell activities occur mainly in the cytoplasm, as we previously mentioned. This is the place where nutrients can be received, processed, and used. And the cytosol, which is just the same thing, cytosol and cytoplasm can be used interchangeably in your textbook and throughout various sources depending on what you're looking at. But the cytoplasm is composed of mostly water plus proteins, carbohydrates, lipid, and inorganic substances. As we have kind of mentioned 3 times now, the cytoplasm serves kind of as the medium or the background in which many of our metabolic reactions will occur. Okay. The endoplasmic reticulum is another organelle that's pretty important for us within the cell itself. And endoplasmic reticulum could be classified as smooth or rough in nature. So smooth endoplasmic reticulum contains no ribosomes on the surface, and rough endoplasmic reticulum does contain ribosomes. Okay, so you can see in your image over here, these little blue dots represent the ribosomes. So this particular section would be considered rough endoplasmic reticulum. This blue section that contains no ribosomes would then be considered smooth endoplasmic reticulum. But basically what this is it's made up of flattened sacs or elongated canals. And the endoplasmic reticulum serves a few different functions. First function is transports molecules throughout the cell through tubular transport system. So the endoplasmic reticulum serves as what is sometimes referred to as the intercellular highway. So it's basically like a highway system that would allow certain molecules to flow around through the cell and be able to travel relatively easy within the structure. The second function would be both protein and lipid synthesis. So again, these ribosomes, if it is rough endoplasmic reticulum, are going to be responsible for producing or synthesizing proteins. And so, the rough endoplasmic reticulum will play a large role in synthesis, again, of proteins and also of lipids. Our third function would be storing and releasing calcium involved in muscle contractions. So, if you have watched the muscle physiology lecture, which was for yesterday, you would have heard about sarcoplasmic reticulum in the muscle. And again, sarcoplasmic reticulum is the endoplasmic reticulum for a muscle itself. And within the muscle, this structure plays an essential role for us in storing calcium. And this would allow us to create a concentration difference or concentration gradient from inside the sarcoplasmic reticulum in comparison to within the cell cytosol or cytoplasm. Okay, so as mentioned in the previous slide, rough endoplasmic reticulum contains ribosomes on its outer layer and functions in synthesis and transport of proteins. And smooth endoplasmic reticulum, again you can see kind of in this area over here, does not contain ribosomes, but it may play an important role in the transportation of lipids. We have vesicles that have a role in secretion, and these are formed by the endoplasmic reticulum as well. And a vesicle is basically like a little pouch or a little backpack that will help us to protect certain items. So again, lipids, for example, would like to be carried in a vesicle because they don't like to be in water, which is basically what the cytoplasm is. Okay, so we've made mention of these ribosomes. Again, this is the site of protein synthesis. Ribosomes will be in charge of creating different types of proteins. The information or like the recipes associated with creating different types of proteins will be stored in the cell nucleus in the form of DNA. So ribosomes contain RNA, which is kind of like the machinery needed to create the recipe. So again, if we're talking about the recipe, let's think of our proteins as different types of bakery items. So we can make like thousands of different types of proteins. We can make oatmeal cookies, we can make brownies, we can make cherry pie, we can make coconut something, I don't know. We can make all sorts of different bakery items. So each recipe is composed of, within the DNA. So the DNA contains all these different recipes for different types of proteins or different types of bakery items. We could consider the RNA as like the machinery needed. So the RNA would be like your mixer that you're using and the convection oven that we're using to make these cookies or whatever it is. Okay, so the RNA is located in the ribosome. Again, we need that machinery in order to produce these cookies themselves, or proteins in this situation. Our ribosomes can be located in two different places. They can be free floating in the cell cytoplasm, and they can also be attached to the endoplasmic reticulum, forming our rough endoplasmic reticulum. Again, as just mentioned on the previous slide, our cell ribosomes can either be number one, free floating throughout the cell, or number two, bound to the endoplasmic reticulum. And the difference with these two is the free floating ribosomes will synthesize proteins that are found within the cell, whereas these membrane-bound ribosomes that are found on the endoplasmic reticulum will synthesize proteins needed for the plasma membrane or for export. So they could be released from outside of the cell if needed. So these are going to produce, again, proteins that will be formed to be a part of the cell membrane, or those that will be released into the world, exported. Okay, moving forward to the Golgi apparatus. The Golgi apparatus is kind of similar actually in appearance to the endoplasmic reticulum. So here's your endoplasmic reticulum over here, Golgi apparatus over here. Again, a kind of a group of flattened sacs. Its job is to refine, package, and transport the proteins that are made from the ribosomes. So again, if you see over here, we've got our proteins that are producing, our ribosomes that are producing proteins. We put them in a transport vesicle. They move over to the Golgi apparatus where they can kind of be packaged up, and then we're going to send them out into the world. All right, so if we think about our whole bakery situation that we've got going, think about this as like Little Debbie's factory, okay? So in the factory, we create the zebra bars or whatever. They're super awesome and delicious. They go in a transport vessel. We send them over to the Golgi apparatus. The Golgi apparatus is going to put them in their little packages and put them in the box and put the price tags on them. Then we're going to secrete them out of the cell. We're going to send them out on a truck to be delivered to all the places in the world. So the Golgi apparatus is going to play an important role in kind of packaging them up and making these ready to go for us. We can release proteins or glycoproteins outside of the cell, and this is the process of exocytosis, in which we release these proteins outside of the cell. Let's see what else. So a kind of take-home message in terms of the Golgi apparatus: it plays a central role in the transport of new molecules from inside the cell to outside the cell, in addition to exocytosis of different molecules as well. So certain things can come in this way and certain things can leave this way. Okay, the mitochondria is our major site for aerobic energy production. So it kind of looks like a mandarin orange within the cell, but within the mitochondria itself, we're going to have various processes occurring related to metabolism. And we'll actually spend a great amount of detail in this course talking about where specifically in the mitochondria the aerobic energy production will actually occur. The mitochondria is sometimes called the powerhouse of the cell. It contains enzymes that release energy from nutrients via aerobic metabolism. And if something is aerobic in nature, we know that this is going to rely on oxygen in order for it to be completed. And the mitochondria actually produces 90% of ATP, which is the energy currency that we use in our bodies. So produces like 90% of the energy that we're going to actually have or use. In terms of the structure of the mitochondria, we have two different membranes. So you have an outer membrane here that surrounds the entire mitochondria, and then we also have this inner mitochondrial membrane, which allows us to have various compartments within the mitochondria, and those will be important for us in terms of the process of energy metabolism that we'll get to again later in this course. Okay, moving on to the lysosomes. So a lysosome is like a little sac within the cell itself, and it contains enzymes that are going to help to break down nutrients or foreign particles. It's basically like a garbage disposal within the cell. So its job is to kind of engulf different things that we're done with, whether they're going to be replaced or we don't need them anymore or something along those lines, and help to break down these foreign particles. Peroxisomes are very similar in appearance to that of a lysosome. It's a membranous sac that's, kind of free-floating within the cytoplasm. They are abundantly located in the liver and the kidneys, and their function is to synthesize bile acids, which will help to break down lipids and also detoxify alcohol. Again, we can understand why the liver and the kidneys would be important for them to have a large quantity of this particular organelle. The liver's job is to kind of process or break down kind of all the nutrients that we're consuming, and also, included in that would be also be alcohol. Sorry, dogs are barking. So again, the function of peroxisomes is to help us break down fatty acids and also detoxify detoxify alcohol as well. Okay, so the cytoskeleton is a network of protein filaments that are found throughout the cytosol, and the function of the cytoskeleton is to maintain and change cell shape and produce cell movements. So again, some of our cells will need to have kind of like a transportation type of method, and the cytoskeleton can allow for this to occur. We can also have the cells change shapes. So for example, when a muscle contracts, the cell becomes shortened, and this occurs through some of the components of the cytoskeleton, right? So the function again is to maintain and change cell shape and produce cell movements when needed. 3 different components or filaments located in this. We have actin filaments, which are also known as microfilaments. We have intermediate filaments, and we have microtubules. And all 3 could be located in a cell. A cell doesn't have to have any of these types, but these are the 3 that are kind of like available to make up a cytoskeleton. Okay, so we have these microfilaments and microtubules that are thin, strand-like items within the cytoplasm. They're going to form this cytoskeleton. They will also allow our muscle contractions to occur. So again, here you can see some of those intermediate fibers and filaments. They could be lying kind of in this configuration, or they could be kind of like spread out throughout the cell like this here, and this would provide us with some structure throughout the cell. Again, actin and myosin are the names of the 2 microfilaments that are involved with muscle contraction. Okay, moving on to the cell nucleus. Again, the cell nucleus houses all of our genetic material and contains the info for protein synthesis. So again, we made the kind of reference to a bakery, and you could consider this kind of like as your recipe box in terms of holding all the different recipes or holding all the instructions for all the different types of baked goods you might be making within your bakery. Let's see, other things associated. DNA, again, is going to direct all cell activities. And we also have some other components that we'll be talking about in the next slide that would be specific to the nucleus. But here you can see the nucleus as a whole, which contains a nuclear pore, DNA, which we said was our recipes, the nucleolus, and nuclear envelope. Okay. So the nucleus, again, is the most prominent feature in the cell, generally speaking, and it contains a few components. So first component that it would contain would be nuclear pores. Let's see if they're in my picture. Nuclear pores. And nuclear pores are perforated channels, and this would allow things to move in and out of the nucleus. We also have a nucleolus or nucleoli, and this is a small dense body composed mainly of the RNA. Ribosomes are actually formed in the nucleolus, and again, remember that the ribosomes then will be kind of like released from the nucleus, and therefore they can, make each of those bakery items that we talked about. So they would contain the machinery needed. So they've got the mixer and the stove needed to bake each of those different types of items. We also have DNA, which is the genetic material. So those are the actual recipes that we contain. So those are the three components of the nucleus. So we have the nucleolus, the nuclear pores, and DNA. Again, what's the difference between RNA and DNA? DNA has the instructions, so DNA contains the original recipe card, where RNA helps to carry out this recipe. So again, RNA contains those machinery types of things that we need to make the recipe. So it's got the mixer and the stove or the oven in order to create the different components that we might need to create. So now we've talked about the various components of the cell. We've mentioned some of the major organelles associated with the cell. Again, keep in mind that not all cells contain each of the organelles, but it's possible to have a variety of these organelles within any given type of cell. We've spent a great deal of time talking about the cell membrane and how it's selectively permeable. Again, this means that some things can pass through and others cannot. Movement across the cell membrane is really important for us in terms of signaling within the nervous system, signaling in the cardiac system, signaling or excitation of the muscles, and also digestive function. Actually, there is movement across the cell membrane for almost every major process occurring within the body. So it's extremely important for us in terms of maintaining normal function. And so we're going to now discuss some of the various ways in which things can move across a cell membrane. So let's start with some various terms here. Diffusion is the tendency of a molecule to move from an area of high concentration to a low concentration. So again, you can see all these molecules over here that are highly concentrated. They're all very close to each other. And basically what happens with diffusion is we want them to spread out so that they're all equal in the distance from one to another. Go. Another factor with diffusion is there is no energy input needed in order for this process to occur. So as previously mentioned in the previous slide, diffusion is a process by which by which molecules or ions scatter or spread spontaneously from high concentration to low concentration. Again, I want to emphasize that these are based on a concentration gradient not an electrical gradient. Oxygen and carbon dioxide can diffuse readily through the cell membrane, and this process is via passive diffusion. Okay, so these items can move through very easily, and they will therefore move from an area of high concentration to low concentration until equilibrium is reached. Okay, so in terms of diffusion through the lipid bilayer, non-polar hydrophobic molecules such as respiratory gases and some lipids, small alcohols, and ammonia can diffuse through a lipid bilayer. Okay? So again, there are various ways in which this can occur. As you can see in the image, this can occur just straight through the lipid bilayer. We can do this through an ion channel, or we can do this through facilitated diffusion. Again, diffusion is an important component for us in terms of gas exchange, absorption of some of our nutrients, and also excretion of some of our wastes. Okay, so again, we've talked about movement of things into the cell. This can also be the process that allows things to leave the cell, whichever the concentration gradient favors. Okay. So most membrane channels are ion channels, and they allow the passage of small inorganic ions that are hydrophilic in nature, again, water-loving things. Ion channels can be selective and specific. They may be gated or they could be open all the time. So you can see here that we have a gate open type of situation. Here you can see the ion channel, and you can see the movement of potassium in this situation flowing through the channel. This gate may be opened via, a variety of stimuli. The gate can also be closed, which would prevent the movement of potassium in and out of the cell. So it wouldn't allow movement through this particular channel. Again, it could be opened or closed depending on various types of stimuli and what the cell wants to really accomplish in this given situation. Okay, so again, here we're having a look at ion channels. Again, they can be gated or they could be open all the time, and therefore, allow items to move through very easily. Again, these can be very specific to particular ions. So in our gated channel over here, only potassium can flow in or out of the cell, and these can only occur when the gate is open, and that maybe would occur based on a particular type of stimuli. Osmosis is the passive diffusion of water. So solutions with a high osmotic pressure are called hypertonic in nature. Again, hyper means above, so hypertonic means that it has a high amount of osmotic pressure. Solutions with equal osmotic pressure are known as isotonic. Again, iso means the same. Tonic, again, we're referring to the solution. And those with lower osmotic pressure are called hypotonic. Again, hypo means below, so this would mean a lower osmotic pressure. Okay, here we can see the various solutions. So again, with an isotonic solution, we have an equal pressure. With hypotonic, the pressure is low, and with hypertonic, the pressure is high. Okay. One other thing to mention with the osmosis that I didn't mention is water can generally pass through a membrane very easily. Doesn't need a channel, doesn't need anything. It can just flow across the membrane without any issues. Facilitated diffusion occurs when substances that are not able to pass through the lipid bilayer get the help of a membrane protein to get across that particular membrane. So carrier molecules are kind of like a revolving door, and they're used to carry large molecules across the membrane, such as certain types of sugars. Fructose is one example of a sugar that moves through facilitated diffusion. And we'll have a look at this. Okay, so basically what happens is this is like a lock and key system. So you have, let's say this is our sugar molecule. It must bind in this specific configuration. Then the protein channel flips over here and opens up to the inside and allows us to dump that particular sugar molecule into the inside of the cell. So kind of the pros and cons with this system, again, it's very selective. It can only allow for the binding of this particular type of sugar into this particular configuration. And it can only move one sugar molecule at a time. So as you can imagine, this process occurs fairly slowly because it takes only one molecule at a time and allows it to flip over, dump that out, and return back to its resting state before another sugar molecule can be moved. Okay, so now we have talked about diffusion through the lipid bilayer. We've talked about diffusion through various ion channels, whether they are gated or just open at all times. And now we've talked about facilitated diffusion and how a molecule must be kind of locked into the middle of the channel. This causes a conformational change with the channel and allows that ion to then move one at a time. You can see that diffusion through this type of channel would be significantly faster than that moving through facilitated diffusion because it doesn't require a conformational change in order to move the molecule across the membrane. A major take-home message related to diffusion is there's no energy input required. Okay, so with any type of diffusion, the major movement is going to be from high concentration to low concentration, and it doesn't require an energy input. So it's free to us in terms of energy investment, we could say. When we look at active transport is going to require some level or some type of energy input. So molecules are going to move against their gradient. So they're going to move from an area of low concentration gradient to an area of high concentration gradient. So our image over here on the left shows us the difference between diffusion and active transport. So again, with diffusion, we're moving from an area of high concentration to low concentration. With active transport, we're doing the opposite. We're moving from low concentration to high concentration. Again, with diffusion, there was no energy needed. With active transport, this requires an energy input in the currency of ATP. So ATP is needed. We're moving things against the gradient. Also, we're going to require a specialized carrier protein or some sort of transport gate. These carrier proteins or transport gate are similar to that of the ion channels that we saw with the various types of diffusion. The major difference is the way in which these items move against their gradient in comparison to down their gradient. Okay. So again, with primary active transport, we required an energy input in the form of ATP. With secondary active transport, we're also using energy, but we're using it in the form of a concentration gradient. So the energy stored in the form of sodium or hydrogen ion concentration gradient is used to drive the other substances against its own concentration gradient. Okay. This can be the form of an antiporter or a symporter. So, secondary active transport can be one of two things. It can be an antiporter or a symporter. It doesn't require ATP, but it does require energy in the form of a sodium or hydrogen ion concentration gradient. And it will secondarily then use that energy in order to move an additional secondary substance against its gradient. So let's have a look at what this actually means. Okay, so with an antiporter, our two molecules are moving in opposite directions, okay? So again, we're using a sodium gradient to move calcium in this picture against its concentration gradient, okay? So sodium is moving down its gradient, so you have a large amount of sodium on the outside. So it wants to flow down its concentration gradient, okay? Calcium is also set up in this configuration. We have a lot of calcium on this side of the membrane, but we want to move more of it against its gradient. So we're going to use sodium's gradient. Sodium moves down the gradient from an area of high concentration to low concentration, and calcium does the opposite. Calcium is going to move against its concentration gradient, so it's moving from inside the cell to outside the cell, from an area of low concentration to an area of high concentration, okay? This is an antiporter because the items are moving in opposite directions. The reason in which calcium can move is because we are gaining energy from sodium moving down its gradient, okay? This could also be the form of a symporter. So with a symporter, we're still using sodium as our source of energy, and now we're going to move something against its gradient in the same direction. Okay, so the name of the porter is based off of whether sodium and that item move in the same direction or if they move in opposite directions. If they're moving in the same direction, this is a symporter. If they move in opposite directions, this is an antiporter. Okay, so again, let's look at our sodium glucose example. So again, we have a large amount of sodium on the outside. So sodium will very easily move from high concentration to low concentration. Okay, we want to move glucose in, but we have a large amount of glucose in, these are G's, inside the cell, and we have a little bit out here that we would like to move. And so glucose is going to move from a low concentration to a high concentration across the membrane. The reason for that is it will move with the sodium and steal its energy from sodium moving down its concentration gradient in order for this process to occur. Okay. A couple other ways in which we can transport items would be through transport vessels. So a vessel is a small membranous sac formed from the budding off of an existing membrane. And this could be from endoplasmic reticulum, this could be from the Golgi apparatus, or this could also be from the cell membrane itself. And a couple different ways or forms in which this can take. We can do endocytosis, and with endocytosis, molecules are engulfed by a portion of the cell membrane and carried into the cell surrounded by a vesicle. Okay, so endocytosis allows us to move things into the cell, and exocytosis is basically the opposite. Exocytosis allows us to get things out of the cell. Okay. A couple different ways in which endocytosis can occur. Again, endocytosis involves things moving into the cell. So we can have pinocytosis. And pinocytosis is when the cell engulfs liquids. So pino means to drink. So this would be allowing us to bring liquids into the cell. Phagocytosis is when the cell takes in larger particles, such as when a white blood cell engulfs a bacteria. So phagocytosis basically means like engulfing or eating. So we're going to take in a larger component or a larger particle via phagocytosis. And receptor-mediated endocytosis is when receptors bind to specific particles and they are then drawn into the cell. Okay, so again, this is related to various forms of communication and signaling that is fairly advanced actually in terms of allowing us to move things in and out of the cell. Okay, As mentioned, exocytosis involves when a membrane enclosed structure called a secretory vessel is formed inside the cell. It fuses with the plasma membrane and releases the contents into extracellular fluid. So with exocytosis, we're releasing substances that were inside the cell, we're releasing them to the outside world. So with exocytosis, we're getting things out. Endocytosis, we're bringing things in.